So welcome to the Biochemistry Clinic, and today we are looking at the urea cycle. So there are just two questions that we need to address today. One has to do with the production of urea, and the other one with ammonia toxicity. Now, when you are asked to talk about production of urea in the liver, I feel it is important for you to mention reactions such as transamination and deamination, because these will give you an idea of how amino acids are converted to glutamate and glutamate to ammonia. Now, when ammonia is produced from deamination of glutamate, alpha-ketoglutarate is also released. But what we have to notice is that this ammonia is very toxic, and therefore the body has to find more favorable forms. And that is the reason why this ammonia is eventually converted to urea in the urea cycle because of its toxicity. Now, when you come to the urea cycle, you need to understand that there are two compartments that are responsible for these steps of the urea cycle. The first compartment is the mitochondria, where we have the following reactions taking place. So the ammonia that has been produced from the amination of glutamate will combine with bicarbonate ions in the mitochondria to produce carbamyophosphate, which later combines with ornithine to produce citrulline. Now, it is this citrulline that is then transported out of the mitochondria to the cytosol for continuation of the steps of the urea cycle. So please take note. These reactions occur in the mitochondria, and after citrulline is produced, it is transported to the cytosol for continuation of the reactions. Now, what other reactions do we have here? So citrulline is then converted to adeninosuccinate. This is in the cytosol, and this adeninosuccinate is cleaved to arginine, and then arginine regenerates ornithine, which goes back to the mitochondria. Remember, at the beginning, we had carbamyophosphate combining with already existing ornithine. This is a cycle. So ornithine is taken back to the mitochondria to begin the cycle. Now, apart from ornithine in this step, arginine also gives out urea, which shows that we've successfully converted ammonia to urea. And this urea is then now taken to the kidneys and given out as a part of urine. Now, there's one important thing that we have to know. These metabolic pathways should have applications, should have a medical importance at least, for them to be meaningful. Now, what happens if something goes wrong with the urea cycle? Now here, I think I would want you to read about the enzyme carbamyophosphate synthase, which is the key regulatory step of the urea cycle. It is the step, it is the enzyme that converts ammonia to carbamyophosphate. Once there is a deficiency of that enzyme, once that enzyme is inhibited, we are likely to experience an excess of ammonia, because ammonia will not be converted to urea. And once you have an excess of ammonia, ammonia is very toxic. And the danger is here. This ammonia can find itself in the brain, and once it gets there, ammonia can stop the TCA cycle by depleting alpha-ketoglutarate. And once the TCA cycle is halted or stopped, we know the consequences. This is like a pathway that gives us the ultimate ATP that we need and things like that. So once TCA is stopped, ATP production is compromised. And then we expect cell death and possible permanent brain damage even. Now, there's some interesting biochemistry of how ammonia does this.